Well, hey there. So, we've been going through the basic pathways of metabolism. And the idea is then that we're taking glucose, 6-carbon molecule, and oxidizing it to generate carbon dioxide and water and extracting energy as we do that. The energy is eventually being converted into adenosine triphosphate, ATP. And that's because as a phosphate donor, it's able to change protein shape and drive reactions. Kind of a simplified term or a way of thinking about it, but it actually works really well as a conceptual notion. So just as a question that was brought up, is when we start listing things of what goes in, what comes out, why aren't we listing the ADPs needed? And I'm trying to keep this rather simple, or as simple as possible. And the limiting factor is never going to be ADP. There's always going to be ADP floating around, because as the cell is growing, as it's carrying out any kind of biochemical processes that are not thermodynamically favorable, ATP is being hydrolyzed to ADP, and so in order for the bacterium to actually need nutrients, it must be using up ATP and generating a free pool of ADP. And so we always have this cycle back and forth. So if for whatever reason the bacteria is in a stasis or in a holding pattern, as it were, the ATP levels may actually be stable and it doesn't eat. But then, as soon as it begins carrying out biochemical activities, it needs a source of a way of rephosphorylating the ADP, and so we begin metabolism again. So, another thing to think about is this system is somewhat indirect in terms of the ADP rephosphorylation, or as they say, ATP synthesis. I disagree with that term. In that, what we're doing is extracting electrons and then using these electron carriers that are holding these high energy electrons to actually drive a proton pump system that in turn is driving the energy for rephosphorylation. And just as a little nerd point, you may see this, there is two ways or two generalized categories of making ATP. You either make it through what's called substrate level phosphorylation. That is the direct conversion of the diphosphate to the triphosphate using some kind of enzyme mechanism. And in mammals and animals, creatine is one of the systems. There's an enzyme called creatine phosphokinase that is able to take creatine, use it as a phosphate carrier, and it can in turn use that to regenerate ATP from ADP. So that's referred to as substrate level phosphorylation, and we see that kind of thing happening in glycolysis, where we make 2 net gain of ATP. You make 4 ATP, but you have to put 2 in. We also see substrate level phosphorylation in the Krebs cycle. There's that molecule of guanosine triphosphate that is synthesized. The term used for making ATP using the proton pumping system is generally known as oxidative phosphorylation. Now, that's really not a great term because oxygen is not involved in the phosphorylation. Oxygen is simply the final electron acceptor. But, back when these systems were being uncovered and being experimented with, people didn't know what was going on, and so they named them the best they could. So what we're going to do is look at some of the different kinds of metabolic pathways now that we have this grander sense going on. And this chapter and this slide set covers a couple of different enzyme models that we've talked about already. And one of the terms that I know I have used incorrectly over the years is the idea of anaerobic respiration. I tend to use that term when we think about in human physiology, when there is a lack of oxygen and we begin using pyruvate as the final electron acceptor, and that's when we generate in humans lactic acid. Plants and yeast generate ethanol using the same process. That really is fermentation. Anaerobic respiration, we're going to get to, that's when you're using something other than oxygen as the final electron acceptor. So in one way, fermentation is using pyruvate as the final electron acceptor, but that specific kind of final electron acceptor, specifically pyruvate, that's referred to as fermentation. When we're talking about anaerobic respiration, we're using other atoms as the final electron acceptor, not oxygen. And we'll get to that here in a moment with a slide. All right, so when we start thinking about metabolism, this is some of the stuff we've been through already. We can put metabolism into this big chart where we've got anabolic metabolism and metabolic metabolism, or anabolic metabolism and catabolic metabolism. So, in anabolic metabolism, our definition was going from the simpler to the more complex. And we're going to require energy, and this is involved in growing. Catabolic metabolism is the exact opposite. It's reactions that are converting compounds and energy into what's needed by the body. It's releasing chemicals and energy to be used during the anabolic metabolism. So polymers are converted to monomers. It is digestion. It is eating. It's what you do to generate the energy so you can carry that reaction out. So if you decide you want to cut one of the reactions out, you really don't have to do the other reaction. If you decide that you're not going to be doing any growing, well, then you don't need to be doing any eating. If you decide that you're not going to be doing any eating, well, too bad. You can't be doing no growing. If you are carefully balancing them, then you're generating the energy you need to build stuff, and you're building stuff at just the right level to maintain where you are. That's basically what weight loss and weight gain and maintaining your weight are all about. So, we discussed further the idea of enzymes and how they are reducing the activation energy of a reaction to allow them to occur at a temperature that is, for us, body temperature, but really it is a temperature that won't denature proteins. Because we're protein-based, because we're built up of these weak-ass bonds, we can't run at temperatures that are too extreme. Now, bacteria, particularly, can live in pretty harsh environments. These extremophiles, they're mostly archaebacteria. They can live at temperatures that would denature my proteins and your proteins, but these are very, very specialized organisms. Most what we think of as mesotherms and the bacteria that are going to be involved in infectious diseases, they are operating at more or less body temperature, and consequently their protein structures are sensitive to excessive heat and excessive cold to a degree. So what we're able to do is reduce the activation energy and thereby allow a reaction to proceed at a much faster rate. The enzyme is not consumed by a reaction, thereby it's being defined as a catalyst. Enzymes are generally proteins, and they have the ability to bind that which they're working on, which we call the substrate. So enzymes are being regulated by a couple of different ways. There may be a time where you don't need to have a specific enzyme because that metabolite isn't around, just as an example. And we're going to be getting into gene regulation, which is a really cool area that I'm particularly keen on as a molecular biologist, a little bit later in the semester. And this is some of the tricks that cells, both prokaryotic and eukaryotic, use to regulate the production of a gene. The production of a gene is actually metabolically rather expensive. And so, you can produce RNA from the DNA template, and that's not too expensive. But when you take RNA and then you turn that into an amino acid polymer, that is metabolically expensive. It requires energy to link those individual amino acids together, plus it requires amino acid biosynthesis, which is not just trivial. So if we don't need an enzyme, it's better not to have it around. If you need the enzyme, you begin synthesizing it. Additionally, there can be enzymes that are present for general metabolic processes, like making these amino acids and things, and you have to have them on probably all the time, but they're not necessarily functioning all the time. They are going to be controlled kind of like a thermostat, like you have your air conditioner or your heater, and you turn the button up on your thermostat to the set point, and that's going to control when the heater turns off and on. Same kind of thing happens with enzyme regulation. Another really cool area of study, that enzymes can be regulated, and we can have an inhibition effect, something that's going to block the enzyme. We can have activation effects. It's going to cause an enzyme to be active. And both these systems are using what is known as a feedback mechanism. And it's exactly like a thermostat. When on your furnace, as we're starting to get a little bit colder now, we won't use the idea of the air conditioner, you turn the furnace on so it's ready. You set the thermostat to 75 degrees, 72 degrees. When the temperature drops below the set point, the furnace comes on. When the temperature rises above the set point, the furnace turns off. The same thing can happen in an enzyme pathway when material that the enzyme needs to act on begins to increase in concentration. The enzyme can be activated. Very often, the product of that enzyme reaction will act as an inhibitor. So as you generate more and more product, you get to a spot where you go, hey, we have enough. That's going to feedback, interact with the enzyme, and cause it to slow up or shut down. So there's all kinds of very cool systems that operate with these sorts of metabolic pathways. So the overall goal is to be as efficient as possible. And this is where we have the idea of enzyme inhibition and enzyme activation. The enzymes can be evolved that they're turned on only in the presence of the substrate. They're active only then. They can be shut off when either there's no substrate floating around or you have generated enough product that you don't need to worry about things anymore, like if you're doing some sort of biosynthetic pathway where you're building something. Once you've made enough of the amino acid tryptophan, you don't need to make any more, and so you can stop. So these are different kinds of inhibitors and activators. This feedback then is a critical part of maintaining efficiency, because it's important that when a molecule is present that you want to break down, you have the enzyme there and ready to break it down. And it's also equally important that when you generate sufficient product in a different kind of reaction, you shut the whole system off. All right. So, as we've been saying, the energy that pretty much all systems, all cells on this here planet use, excuse me, are the nucleotide triphosphate. And this is specifically here, let me get my little pointer. This is ATP. So here's the adenine group, or the adenosine group with a nitrogen in it, nitrogen-containing base, as it's known. Here is the ribose sugar, the 5-carbon sugar. It's got 2 hydroxyls. This one is always there. This one, if it's removed, it is said to be deoxygenated, and then you have a deoxyribose. That's what makes up DNA. And then these are the phosphates. So you've got 3. The high energy bonds are right around here. They are bonds that have a lot of energy in them, and when they break apart, they can transmit that energy. Additionally, the phosphate and its giant negative charge also can affect things. So when we talk about ATP going to ADP, what we mean is the triphosphate is losing that phosphate and becomes the diphosphate, and that energy can be used. So rather than having fire that's somewhat uncontrolled and a big blow up, we have very small little pops of energy, and it's much more easily controlled and easier to build. So that's why we have phosphate. I still think about, just recently there was the discovery that there are vast underground bodies of water on Mars potentially, which would be a really cool place for life to exist. I suspect if we find life on Mars, it is probably going to be using some kind of phosphate molecule to drive chemical reactions. I would imagine that if it doesn't use phosphate, it's going to be so bizarre that we may not even recognize it as life. This mechanism is so ubiquitous on the planet. Everybody, all creatures, great and small, plants, animals, bacteria, everything is using phosphates. So another part of the enzymatic idea is you want to have little stepwise changes in a molecule, because it doesn't take a whole lot of energy to make that small change. So you're not going to take a molecule and completely rearrange it in one throw. One enzyme can't do that. It can do simple individual changes. And at the same time, when you're breaking stuff apart, just like we saw in glycolysis, you don't just set it on fire immediately. You break it apart a piece at a time, pulling one carbon off at a time. Each time you pull the carbon off, you get a little bit of energy. And rearranging bonds gives you a little bit of energy. So fire is very rapid oxidation. Metabolism is very slow, stepwise oxidation. Another side part of this is you build these little intermediates each time, and each one of these might be a branch point for other molecules. So the system can get very complicated. Here's an example of taking the amino acid tyrosine and turning it into the pigment melanin. And it's done in 6 steps, in which the changes are really very, very small. So if you can track them here, we've got the addition of a hydroxyl here. We remove the hydrogens here and make oxygen double bond. We then are going to remove the hydrogens from this nitrogen and cause this ring structure to close up. The hydroxyl groups are added back on, and then they're removed, so back and forth, and we end up with this product called melanin, which is a pigment. It's just an example. This, I think, is a hilarious picture in which we can trace all the different metabolic pathways in an organism, in a cell. Pretty darn complicated, yeah? Just so you know, this is the Krebs cycle probably here as a main trunk line. This is a very complicated chart of metabolic pathways. And what you're seeing is the fact that we have to be able to break things down, and bacteria in particular, they can build every single component. There are bacteria that don't require large molecules. They only require a source of carbon and a source of nitrogen, and they can build it all. You and I, we're not nearly so lucky. We have to take in proteins because there are a set of proteins that we can biosynthesize, a set of amino acids, and then there are a set of amino acids that we can't biosynthesize. So those amino acids that you can't biosynthesize, you have to take in through food. And another example, something I've been actually kind of working along, is vitamin C, which is a really critical cofactor for a lot of different systems. There are organisms that can biosynthesize their own vitamin C, and then you and I cannot. And these biosynthetic pathways are controlled by individual genes, and they're all part of this grand scheme. So some organisms are going to have a somewhat simpler biochemical pathways because of what they can biosynthesize. Other organisms are going to have a really complicated biochemical pathways, because they can biosynthesize everything. It's all pretty complicated, but this is just flat-ass ridiculous. Go ahead and start memorizing this. No, don't. All right. So, just as we said, the reactions of all these different processes, they are often used as branch points for a lot of other things. So when we're looking at glycolysis, we take glucose and begin breaking it apart. That 3-carbon molecule can be used to generate DNA and RNA. We take pyruvate and put that into acetyl-CoA, pop off the carbon. We can make fatty acids 2 carbons at a time. We can also break them down. We had the citric acid cycle, Krebs cycle, and each one of those intermediates along that Krebs cycle can be used as branch points for making a bunch of different amino acids, lots of things. So it is not just a simple sort of breakdown. It is a complicated thing depending on what the organism needs. All right, so this summarizes pretty much what the pathways we've been talking about. Glycolysis is taking sugars primarily and makes a little bit of ATP, 2 molecules net gain of ATP. You extract high energy electrons by putting them onto NAD, and you end up with pyruvic acid. The Krebs cycle is taking pyruvic acid, pulling off a carbon, oxidizing it as CO2. It makes nucleotide triphosphate. It's moving more electrons around, and it is where carbon dioxide is being released. Then finally, the electron transport chain is converting the energy in those electrons into the energy required to pump protons out. That energy is being used to actually add a phosphate to ADP to make ATP. And the electrons have to go somewhere, and in aerobic metabolism, they go on to oxygen. And you generate a molecule of water. So this is way more detailed than you need. If you decide to go into medical school or take biochemistry, yeah, you may have to memorize these individual steps. Happily, nothing is bigger than 6 carbons, and in fact, very quickly, right there, it ends up being all 3 carbons, glyceraldehyde phosphate. So the number of carbons you have to deal with isn't all that high. Then when you get into the Krebs cycle, you're taking the 2-carbon acetyl-CoA, putting that into a 4-carbon molecule, making citrate, which is a 6-carbon, and then you start losing carbons again. So the biggest molecule you got to worry about is the 6-carbon citrate molecule, a 6-carbon glucose. So it's not really all that hard to memorize the structures, but we ain't going to do that. Don't worry about it. The main thing I want you to understand is what's going in and what's coming out. So glycolysis is converting glucose to pyruvic acid. We generate 2 ATP, and we generate the 2 NADH. And now we have all these different kinds of metabolic intermediates that if you do need to make other things, you can do that too. The Krebs cycle, it's also known, I think I mentioned, as the TCA cycle or the citric acid cycle. TCA stands for tricarboxylic acid cycle. I'm not sure who developed that term. I always thought TCA meant the citric acid cycle, but it ain't. So, somebody here didn't get Christmas cards from Hans Krebs and they got aggravated, so they're not calling it the Krebs cycle. It's the Krebs cycle. Hey, I give Hans his due, man. The guy was smart. Call his name out. It's the Krebs cycle. We've got the NADH is being generated here. I said lots, 3, whatever. It means that we have to have a fair amount of NAD there. We make this nucleotide triphosphate. We make a reduced FAD, or FADH2 as it's known, and we generate carbon dioxide. And so we've got these multiple spots. Citrate coming all the way around to generating the 4-carbon molecule oxaloacetate. The last step is the electron transport chain and oxidative phosphorylation. And this is where we have the ability to reoxidize electron carriers. And so NADH and FADH2 are dumping their electrons on. So we're not showing the complex here where FADH2 is being used, but it is. The protons are being pumped out, and then they can come back in through this protein that is known as ATP synthase. And as I said, it should really be called ADP rephosphorylase, but they never asked me. And there are a variety of different intermediates, things like ubiquinone and cytochromes that are actually helping to pass the electron from one carrier to the next. So ultimately, the electron is going to be used to reduce a molecule of oxygen, of O2. And this is the way we can think of it. Rather than calling it O1, chemists demand us that we call it 1/2 O2. Okay, fine. It is a diatomic molecule. O1 doesn't exist very long because it's so chemically reactive. It is the bleaching agent in peroxide. So you generate an O1, and then the other oxygen floating around is going to be used to make another molecule of water just right afterward. Yeah. All right. So now, this is the idea of anaerobic respiration, which is anaerobic respiration is where we're going to carry out everything the exact same way, glycolysis, Krebs cycle, electron transport. The only difference is instead of using oxygen as the final electron acceptor, we're going to use something like a sulfate molecule, or an iron molecule, or a nitrate molecule, or an NO2 molecule. And one of the things that makes these guys a little bit different is that oxygen has got a really big draw for electrons. You remember back in the very early days of our discussion, we were talking about bonds. And hydrogen bonds happen because oxygen is electronegative. It's an electron hog. And it tends to hold the electrons around itself, making it so the oxygen has a slight negative charge and the hydrogens on either side have a slight positive charge. And that's what makes hydrogen bonds. Oxygen's ability to pull electrons, its electronegativity, has a greater drawing power, and it just generates a little bit more of a pull, which allows the different kinds of systems there to pump a bit more hydrogen and work a little bit harder because the electrons have a little bit more energy. And consequently, aerobic respiration really is the way to go because you get, per NADH, you get around 3 ATP. When you compare it to using not oxygen, but sulfate or iron or nitrate, you end up only making 1 or 2 ATP. But the advantage is you don't need oxygen, so you can work in really weird environments, but you're not going to be making as much ATP, not as efficient. You got to work a little bit harder to get nutrients and things. So, now, when we are going to use pyruvate as the final electron acceptor, that is anaerobic respiration, but this is a very specific type of anaerobic respiration that we call fermentation. So fermentation is where we're going to use, the title says organic terminal electron acceptor. Pretty much it's going to be pyruvate. And we do this because in order to carry out glycolysis, you have to have that NAD molecule. NAD is typically being generated or regenerated by using oxygen to dump the electron off of NADH and regenerate it. If there's no oxygen, and you can't carry out anaerobic respiration like you and I can, I just can't use sulfur or nitrate. You have to carry out this idea of fermentation, where we're going to use pyruvate as the final electron acceptor. Now, why is this cool? Because when you take pyruvate and you reduce it, you add an electron onto it, you can make all kinds of really cool molecules, carbon-based molecules. You got 3 carbons, you can play around with them. You can make things like lactic acid. You can make a molecule called acetaldehyde that can then be used to generate ethanol. So in this spot, what you and I do is we take pyruvate and just use that as the electron acceptor, dump NADH onto it, the electron, and generate lactic acid. Yeast and plants are popping a carbon off using an enzyme that we don't have to generate a 2-carbon acetaldehyde. That is the place where the electrons are being dumped, and you end up making ethanol. All right, so I guess to say that pyruvate's the final electron acceptor, it's not exactly, but close enough. Conceptually, this works. Thank you very much. So, you make a whole range of fermentation products. Ethanol being one, but butanol, propanol, acetic acid, propionic acid, which smells a lot like somebody just threw up, by the way. Ammonia, all kinds of different things are generated by fermentation. And humans have learned to use fermentation products. There are bacteria that are producing propionic acid and generating Swiss cheese. There are bacteria and fungi, Lactobacillus and Streptococcus and Aspergillus, which is a fungus, that are able to generate lactic acid. And this makes things like soy sauce and yogurt. The yeast Saccharomyces is able to generate ethanol. Clostridium generates acetone, essentially nail polish remover. It also can make isopropanol, rubbing alcohol. Let's see. Acetobacter is able to generate and make acetic acid. make vinegar. So you can take apples and make cider vinegar because you're either adding the bacteria or the bacteria is naturally occurring there and you just let it go. And all of them are building these molecules as a way of dumping the electron from NADH to regenerate NAD. So all kinds of products that humans have developed that uses fermentation. Now, we're only going to take a moment to think about photosynthesis. It's actually a really cool kind of mechanism in which we have pigments that are able to absorb light. And when these pigments are hit by light, an electron goes into a higher energy state. It is essentially a same kind of electron energy that NADH can deliver to an electron transport chain. It's just that now you do it by light. And that electron then passes through a electron transfer or electron transport chain. And like all electron transport chains, you use that to make ATP by generating this proton gradient. And we have a range of different kinds of photosynthetic mechanisms in bacteria. In plants, we are splitting water to form oxygen and to extract the hydrogen electrons from the water, essentially. You're oxidizing water. And that is an oxygenic kind of photosynthesis. You generate oxygen. Now, there are bacteria that are called anoxygenic, and they don't use oxygen. They use hydrogen sulfide and other kinds of organic molecules. And so we've got a couple of different kinds of systems here. And what you're looking at is where the electron is coming from. That's the really important point. In the metabolism pathways we've been talking about, the electron is coming from glucose. Here, the electron is coming from water or hydrogen sulfide or different kinds of molecules. So it is a way of generating ATP from light energy. They don't need to eat in the same way that we do, anyway. We have a couple of other oddballs, the chemolithotrophs. These are organisms that actually eat minerals. They don't eat, they're not taking the electrons from organic compounds. So we have things like sulfur bacteria that take hydrogen sulfide and generate sulfate. They actually, whereas we dump out water, they dump out elemental yellow sulfur, which would be pretty weird. We have nitrogen-fixing bacteria that are able to take ammonia and generate nitrate, a pretty important soil bacteria in the agriculture business because they're generating nitrate that can be used by plants. Nitrobacteria is using different forms of nitrate. And iron bacteria, they're taking iron and using and oxidizing iron to a different oxidation state and pulling the electron out. In each one of these steps, the bacteria are oxidizing this input and pulling the electrons out. The electrons then can be used to generate carbohydrate from a carbon source, probably CO2. They also can be used to drive electron transport chains to make more ATP. So altogether, we've got this interesting kind of system. All organisms are going to require an energy source. Now, for you and I, that's an organic compound, but it can either be a chemical source, which is a rather general term, or it can be light. And so we have the phototrophs. Their carbon source is either organic compounds or it's carbon dioxide, like plants. And they're using water to reduce carbon dioxide. The electron is coming from water, either the oxygenic or the anoxygenic. Now on this side, we're using various chemicals to generate the carbon source, and you can use carbon dioxide, and then these are some of the rather odd bacteria that can do this. For us, or at least for our purposes, the pathogens that are non-viruses all come down this side, and this actually is pretty much all living pathogens, not just bacteria, but liver flukes and parasites and fungal infections. They are all down this side in that they're taking organic compounds, what we would call food, and the final electron acceptor is either oxygen, and that's what we do, and most pathogens, or it's not oxygen, and it's either an organic compound or an inorganic compound. And get that block out of the way. Oh well. All right. So some of these guys are things like Streptococcus, which can be pathogenic. Clostridium botulinum uses inorganic compounds, and so most pathogens are over here, but some are actually doing some odd things. There are only a handful of pathogens that we would consider to be anaerobic. Most pathogens are aerobic. And just as a way to think about this, anaerobic pathogens are going to be guys that are going to be living in an anaerobic environment, and so a deep puncture wound might have the ability to become anaerobic way down the bottom of that hole, the nail puncture or the dog bite, and so you can get some anaerobic bacteria growing in there. Parts of your gut can be anaerobic, so it's not real common to have anaerobic spots. If your sinuses get completely blocked up, your sinuses can go anaerobic, and they can begin generating an atmosphere or an environment that is capable of growing these kinds of pathogens. But most pathogens are going to be growing in an aerobic environment. So things in the lungs, things in your skin, things in your blood. So there's not a whole lot of anaerobic pathogens that we have to think about. Most are aerobic. All right, so that is Chapter 6. And we're going to be getting into some of the gene regulation and things, some very cool stories here, and see where we go from there. All right, so we'll pick you up on Friday and see what comes from that. Thanks for your attention. I'm going to post all this stuff, and I'll talk to you later. Peace out.